Hi, welcome to a virtual tour of the Organic Robotics Laboratory at Cornell University. My name is Emily Baker, and I am a graduate student in the Mechanical Engineering Department. In this video, you will see a wide range of different robots and robotic applications that we work on. Our lab draws inspiration from nature to better understand nature and to gain knowledge about how to make better robots. This video will focus on fish robots, amphibian robots, jellyfish robots, replicating snake skins, and 3D printing human heart arteries.
Hello, everybody. My name is Cam Aubin. I'm a fifth-year PhD student at Cornell in the Department of Mechanical Engineering, and I'm going to talk to you today about robots. So let's get started. I'm going to shrink my screen here a little bit. I'll be up here in the corner. So let's talk a bit about robots. When you think about robots, you might think of something like this guy I have here on the screen, like sort of a metal gizmo with arms and wheels. You might think of a drone, like an aerial drone for photography, or you might think of like a car or plane, train, automobile. All of these things are kind of robots, and they all have something in common, regardless of what they look like. They have a power system. They have some amount of control, something to control what they do, and they also have actuation. An actuator is just a fancy word for something that makes something move. So like a motor is an actuator, an engine in your car is an actuator. There's lots of different types of actuators. And so robots have all of these things, and they're all kind of connected and daisy chained together. You have a battery that controls an actuator, that controls the robot's muscles, or you get the idea. And there's a bit of an inefficiency in the way that robots are made, because you have all these specific parts and they all just kind of fit together, but they're all only really good at doing one thing. And in my research, I like to think of what would happen if we combine these parts into one integrated system that did all of these things together. What would that look like? Something like a battery that's also your motor, but that also controls the robot. Like, what would happen if we kind of merged all these things together? We would, in theory, get a robot that's more multifunctional. It can do more functions. It can do more things. It works better, and it can adapt and change. And the inspiration for this is kind of the human body, which, if you think about it, is sort of the most multifunctional machine, at least that I can think of. Human body can do lots of different things, and the organ systems inside of us can do lots of different things. Let's talk about your blood and your circulatory system. Your heart pumps your blood throughout your body. It collects oxygen from your lungs, your respiratory system, and it transports that oxygen to your muscles, your musculoskeletal system, and that allows you to move. It also transports nutrients throughout your body, so you can live, right? And it takes care of waste. It's involved in your immune system. Think of white blood cells, right? And fighting off diseases. So your circulatory system does a lot of things, and it's also deeply connected with all of your other systems in your body. It's like this big web, this big network inside of your body. And our idea was to sort of take this line of thinking and combine it with a robot. Like, can we give a robot lots of functions by really integrating its systems, by making them all, together, all one system? And so we did that. We created a robot, fish. We created a fish robot. And the paper that this was based on is called Electrolytic Vascular Systems for Energy-Dense Robots. It's a bunch of words there that ultimately amount to this. We created a robot, and that robot has a liquid inside of it. That's what you can see in this fish, these dark blue patches right here. There's actually a red liquid inside of it that is a battery. It's a liquid battery. And so, being a battery, it supplies energy to the stuff inside of the fish, the control computer inside of it, and the pumps and the motors and all that stuff. But the other cool thing that it does, it doesn't just supply energy, it actually is the thing that actuates or moves the fish. When you pump this robot blood around the fish, because it's made of soft materials, parts of the fish expand and contract, and you get the movement of the different fins. And this whole system is really cool because we have this energy spread out throughout the body of the fish, so it allows all the different parts of it to move, but it also, instead of just having the energy in one space like a single battery, it's all spread out, so we get lots of energy, and that allows us to operate for up to 36 or 37 hours, which is totally crazy if you think about it. That's like a very long time for a robot. If you think this fish looks kind of weird, we based it on a real fish. Kind of. It's based on a lionfish, which is an invasive species of fish that you can find like off the coast of like the southern United States, North Carolina, that kind of area. It has lots of ornate fins. It's got spines on it. It's kind of a nasty fish. And that's why our fish has this kind of crazy looking mohawk here. That's like its dorsal fins here. So this is a diagram of the robot. There's two battery systems inside of it. One is the tail. There's a left and a right side of the tail. And as fluid is pumped from one side to the other, one side will expand, and then you can reverse the process, and the other side will expand, and you'll get tail movement back and forth. Similarly, we have these dorsal fins that are connected to pectoral fins, and as the fluid from there, which again, it's all providing energy, it's a liquid battery, as that fluid is pumped from the dorsal fins to the pectoral fins, the pectoral fins fan outwards. And so you actually get fin movement there too. And this is all done using different pumps and a computer inside of the robot. This is just a diagram of it. This is like a top-down view of the tail. And this is just kind of showing you what I mean. This red stuff right here, that's the fluid. And as it goes from one side to the other, the one side gets smaller, contracts because there's less fluid inside of it, and the other side expands, and you get that bending that we see. And this is not super important, but it's just a diagram of the chemistry happening here. Basically, the fluid, this robot blood, is filled with ions, just like all kinds of batteries are. You've heard of lithium-ion batteries, right? Well, this is a zinc iodide battery. It's got zinc ions instead of lithium ions, and it's got iodine inside of it. And these ions have charges, and they just basically move around, and the movement of charge is what's generating electric current. So it's generating the electricity that powers this fish, which is pretty cool. And so this is a video of the tail moving. So it's underwater, obviously, so that's why it's a little blurry. And we filmed this all in a fish tank in our laboratory. And it's not super fast. This isn't a super fast robot, but we didn't set out to make a super fast robot. We set out to make a really energy-filled robot. Again, this can work for over a day and a half. This is the pectoral fins moving. You can see how they kind of push outwards. So the fluid comes from the dorsal fins to the pectoral fins and pushes them outwards. And this is what lionfish actually do to communicate. They fan their fins at each other as like a sign of aggression or to tell other fish to do things. You know, they're very territorial, actually. And then finally, we have some of the inside right here that I can show you. This is our microcontroller. This is the computer inside of the robot. And we can control that using Bluetooth, so we can actually wirelessly talk to the robot and tell it to move around. And this is a video of that battery fluid moving through the robot. We call that a catholyte. You know that batteries have like a positive and a negative end. Well, the positive end is called the cathode, and so a liquid cathode is a catholyte. So, fun fact for you guys, right? And then finally, here's the video that you wanted to see. This is the fish actually moving through the water. So again, it's very slow, but you can see the tail fin. You can see the pleats in the tail moving here. It's a little slow, right? It moves at about 2 feet a minute. The fish itself is a little over a foot long, but because it can operate for over 36, 37 hours, it can actually move almost a mile in that time. It moves like 4,000 feet plus, which is kind of crazy. So this is the type of technology that I'm interested in doing research in, because I've combined functions here. I've taken energy storage as a battery, and I've also made that battery be the thing that makes the fish move around. And so, yeah, I hope you enjoyed this presentation. I hope this is interesting to you guys, and I hope you enjoy the other presentations from my colleagues. Thanks.
Next, I will be presenting Autumn Pratt's and Shu Liu's work on a jellyfish robot. The jellyfish robot has paddles surrounding its body. These paddles are made of a soft, transparent plastic, similar to that of garbage bags, and have channels that run throughout them. When the channels are filled with a liquid, the paddles become stiffer. The change in stiffness allows the paddles to push water, enabling the jellyfish to swim. In this video, the jellyfish moves from the middle of the tank to the other side of the tank, and the parts of the robot that you see moving are the paddles. Another interesting feat in this robot is that the fluid is an electrolyte, meaning it's being used like a battery to power the robot. So not only is the fluid being used to generate motion that allows the jellyfish to swim, but it's also being used to power the robot. Another project Autumn Pratt is working on is an amphibian robot depicted here. Living part of its life in water and part of its life on land, the amphibian robot has a tail fin for swimming and legs for walking. The body of the robot is 3D printed from a transparent plastic that allows you to see the internal electrical and mechanical components of the robot. Additionally, the inside of the robot is pressurized, allowing its legs or tail to be inflated. This video is demonstrating the robot's ability to swim in water. Here, a tendon is moving the tail fin to produce a swimming motion. A tendon is a piece of string that is on one end connected to a tail fin and on the other end connected to a motor shaft. The motor is programmed to spin, reeling up the string or unreeling the string. By pulling on these tendons that are connected to the tail, motion is generated in the tail and the robot can swim. This video demonstrates the tail fin being pulled in. This is accomplished with a tendon that gets pulled in with a motor. The tail fin has been pulled in, and the pressure that was once in the tail fin has been redistributed to the legs. With the pressure in the legs and the tendons inside the legs, the robot is able to move its legs and scoot its body across the table. In summary, the amphibian robot can swim with its tail fin or walk on ground with its legs, with the clever use of pressure, tendons, and shape changes.
Hi everyone, so I'm super excited to be talking to you guys today. So I wish we could be in person, but obviously this will have to do for now. My name is Rachel Miller and I'm a PhD student in material science and engineering. So even though Rob's group is in mechanical engineering, it's cool because as a material scientist, I have lots of room to move around. So my path within material science is mechanical engineering. And I've been working on a project that sometimes I like to call the snake that can't be sniffed. So first of all, we're going to look at this video. Here is in red highlighted a image of this snake. Now this is a nighttime video and this porcupine just runs right over that snake. So what's weird about that is that, so in this nighttime video, like I said, this snake is just laying there doing its thing. And normally this porcupine would eat it right up. In the daylight, it would easily be seen and become breakfast or lunch. In this case, it would have been dinner. But this snake is a little bit different in that it can't be smelled. We didn't really know why. But we wanted to find out why. And so this was actually the first land vertebrate, or basically large land animal, to be known to be chemically cryptic or undetectable by smell. So this is what this guy looks like. And like I said, we wanted to find out why that was. This was originally actually brought to us from some ecologists or animal scientists over in South Africa at the University of the Witwatersrand in Johannesburg. So what's cool is that observed in the wild, these snakes are, again, undetectable by smell, by predators, prey, or even each other. So it wasn't like they were chemically invisible or smell invisible to just their predators or just things they wanted to eat, but also to even other animals within their own families, their own species. And then also in a controlled environment, these researchers took swabs of their skin and they were undetectable to elephants and trained dogs and meerkats. Fun fact, both dogs and meerkats are used to do these kinds of studies. And so there was a lot of evolutionary reasoning behind why these snakes are undetectable to smell. They like to sit and lay around for up to 3 weeks. Before they even catch their prey. They will literally wait for their prey to come within their striking distance. And they're, as you can see in that other porcupine video, they're very good at just laying around, being very still, even when a predator approaches. So this evolutionary reasoning, the way they observed this behavior in the field, and then also these controlled lab experiments led us to believe that this puff adder has this really unique ability to not be smelled. But we couldn't really figure out why, or at least a lot of the more animal science people didn't really have a great answer, but they had a direction, which was one of the only variables left to test would be the actual surface of the skin of the snake. Which is actually very surfaces and things like that are really popular and really important in material science. So they approached us as a group that does both mechanical engineering and 3D printing and material science to maybe understand better the surface structure of the snakes and their skin and see if there was a way that the odorants or the things that smell were interacting with the surface. So there's lots of different hypotheses that we have, but one of them is that see these little microbes or these little things that are like bacteria, mainly bacteria and fungi or fungi, which are things that ultimately give off things that smell, things that give off odorants, are being brought down into the porous part of the skin away from what are called the fingers or the little ridge structures, which I'll have pictures of those later. And so then what happens is that if you can bring all of those microbes or the things that produce the stuff that smells away from the surface and into the pores of the skin, then you can actually have air pass over the skin and not pick up any of those odorants or those things that smell. So, like I said, we hypothesized that there's these surface structures or these fingers that interact with the microbes or the odorants, the smelly stuff, or rather they don't interact with them. And then the air doesn't smell. So the air around it, air around the snake doesn't smell. But in order to test this hypothesis, we have to separate out lots of different variables. So the snake skin itself is made of something. So it's made of something called keratin, stuff that's in your hair and in your nails. And that's like another compounding factor to talking about the geometry or the structure of the skin. The environment, so we can't just take a whole snake and like do the experiment again because we'd be doing the same experiment. So what we wanted to do was completely just isolate the geometry or the surface structure of the skin from everything else, from the environment, from the snake itself, from the material. And so that's actually really tricky, but the way to do that is with 3D printing. So, but you'll think, how the heck do you get these tiny features of skin with 3D printing? Or how do you even start to begin to think about 3D printing snake skin? So what we do is we take the shed skin from these snakes, take a little piece of it, and this is our sample. Think about this as our sample. And we put it in what's called a 3D nano X-ray computed tomography scanner or a CT scan. So you may have heard of a CT scan, say if like you or someone you love or someone you know has like a brain injury or some other kind of soft tissue injury. What some of the things that they'll do is maybe an MRI or a CT. So normally if you were to get stuck into a CT scanner, all of the detectors, this giant thing would rotate around you. But with these tiny samples and with big x-rays, what we do is we just stick the sample on the little stage there. We spin around the stage. And while we spin it around, we shoot x-rays through the material. And so at every single cross section, as it rotates around, we can start to build a three-dimensional image of whatever we're shooting x-rays through. So like I said, we take a little chunk of the skin and throw it in the CT scanner, and this is the kind of image we get. This is actually three-dimensional, but a 2D version of what the three-dimensional picture, or they call them voxels, three-dimensional rendering of what we create. And then we can actually go even smaller than that. So take that section and zoom in, and we get to see in all of its glory, all of the detailed fingers and structures of the snake skin. So this is super cool. It's probably one of my favorite parts of this project, of working with this stuff, is that I get these beautiful images, these beautiful three-dimensional renderings to create what are called STL or .stl files, which are then 3D printable. So I could, but the problem is if you look at the scale bar over here, this is super small. So your traditional 3D printer is not going to be able to print this. But we have a way to do that. So what I do is I slice off the edge using a software called Mesh Mixer, and I array together all of the sections to make one big sheet. And I put those sheets together to make an even bigger sheet. And then I use what I'm going to call a tiny 3D printer, but it's actually kind of big, but I use it to make tiny things. So I throw the STL file into what's called a nanoscribe, or nanoscribe GT2 is the official name of it. And you can get nanoscale resolution 3D printing. So this 3D printer is able to create structures that are even smaller than a fraction of the width of your hair. It's pretty crazy. Using completely different technology than most traditional 3D printers. Again, cool material science. I'm able to actually get one of my sheets. So again, this is pretty small. For reference, this is a little gel pack or a little holder that's about yay big, about like an inch or so by an inch. And this is my little sheet. So what's cool is, as far as I know, this is the first 3D printed snake skin ever created with its full resolution. So what's next is I'm going to be sending these samples once hopefully things like COVID aren't really influencing the world as much. I'd like to either go or send my samples to South Africa to be sniffed by the meerkats and see if it's really just the surface geometry that's causing all of these odorants to not be smelled, or maybe if it's something else. But anyway, I'm super excited that I got to show you this video, and have a great rest of your day. Bye.
Hi, my name is Emily Baker, and I'm going to be talking to you about my project on arteries. Arteries supply the heart with blood and oxygen. However, plaque can start to build up along the walls of arteries and can restrict the blood flow, and in severe cases, block blood flow. This disease is known as coronary artery disease, and it's a leading cause of death in the United States. The image depicted here is of 4 arteries, with the one furthest to the left being a healthy artery. As you move towards the right, plaque starts to build up along the walls of the arteries. You can see how this is starting to restrict the blood flow, and in the last case, the blood flow is completely blocked. In this project, we want to be able to help doctors more accurately determine when a blockage is dangerous to a patient and to help recommend the most practical procedure for the patient. For example, one such procedure is called a bypass surgery, which is depicted here. This is where an artery is taken from another part of the body and is attached above and below a blockage in the artery. This then allows the blood to flow around the blockage. To help doctors, we are 3D printing models of arteries and analyzing the mechanical properties and fluid flow. In order to make models of arteries that will help us understand how they perform inside the body, we 3D print them. The picture on the left is a CAD model of an artery made in software. This model then gets uploaded to a 3D printer. The picture on the right is a carbon M1 3D printer. This 3D printer works by filling the resin tray with a liquid resin. Then the build head moves down into the resin tray over a window that is located at the bottom of this tray. This window allows light, specifically UV light, to pass through it and onto the build head. The UV light causes the resin to form a solid in a layer by layer process. The challenges associated with 3D printing an artery, similar to those found in the human body, is that the material must be very soft. Commercial 3D printers lack the ability to print these soft materials, so we make our own. In summary, we are trying to replicate the behavior of healthy and plaqued arteries to help doctors treat coronary artery disease. We hope you enjoy taking a virtual tour through the Organic Robotics Lab. We hope to see you in person next year.